This is a brief review of fluorescent microscopy for those of you who are following our, our goal in staining the EPS in the biofilm. So as you saw from the prior videos, it's very difficult to detect the EPS in a biofilm. And what fluorescent microscopy does is it stains proteins, lipids, and carbohydrates in the EPS. So this is our staining station. It's actually a fairly simple procedure, fairly easy to understand. We put a biofilm sample on a slide and then stain it with these fluorescent dyes. In this case, we're using a live dead stain just as an example, so you can gain a sense of what this imaging is like. The stains are kept in ultra cold at minus 20 degrees centigrade, and they're kept frozen and then unfrozen when they're ready to use. And then they're imaged in a fluorescent or confocal microscope that excites the dyes. So this is one of the biofilm samples, and this is what it looks like. You can actually see individual bacteria stained green. This is live dead stains, so the dead bacteria are red. You are not seeing any biofilm EPS matrix here. This is just an example of showing how the fluorescent dyes work. And this is a biofilm grown on a glass slide, and you get a sense of the number of bacteria present. And here you can see another sample and the individual bacteria that are evident. This is a live dead example, and you can see bacteria that are both alive and dead in the biofilm sample. So we'll be using different stains to attempt to make the EPS matrix visible using confocal and fluorescent microscopes. Just out of curiosity, I did take a diamond and split the glass slide and mounted one of the specimens on a stub for electron microscopy viewing, so you could get a sense of what the stains were actually showing. This is the fluorescent stain. This is the SEM of the exact same sample. And you get a sense for how to correlate the SEM findings with the fluorescent microscopy samples. And here you can see the individual bacteria and also quite a bit of EPS. So we're attempting to visualize with fluorescent microscopy the EPS. So this gives you a little background in how these images will be.